Okay, welcome back. So I'm going to talk through a part two of the respiratory system. So hopefully you've got a sort of few bits in your workbook where you've kind of noted the structure of the main respiratory tubes. I've also given a little bit of background on the nasal cavity. So the presentation starts with a couple of bits of that, but I'm going to sort of rattle through them fairly quickly. So, just a quick reminder, what we're working towards is this bit here, which is describe and explain the function of the respiratory system. But the it's important to see the how the structure of the system enables that function. So we're describing the structure and function of the respiratory system, and importantly, we're sort of expanding that to the merit to explain really how that structure kind of allows that function to happen. So um, we sort of started with some we finished with some questions last time. So if you just want to have a look at these, pause the presentation and just quickly answer them. And then if you remembered this lot, then well done. Just pause the presentation, just have a quick look over those. And just a quick recap. So we looked at our various sections. So we considered the difference between the upper respiratory tract and the lower respiratory tract. We looked at the nasal cavity, we looked at the conchae within the nasal cavity. We talked about the structure of the trachea, the C-shaped rings of cartilage. We looked briefly at the bronchi, these complete rings of cartilage, all lined by this sort of ciliated epithelium. So the cilia, those little hair-like projections, which sort of try and allow sort of removal of debris and dust. We saw how the bronchi divide into bronchioles, and their diameter decreases as they get there. And we saw a slight distinction between the left and right lungs. So you should hopefully sort of start getting a nice sort of picture of what that respiratory tract looks like. We looked very briefly at the sort of process of gas exchange. We looked at this picture as we saw the scheme of circulation in that oxygen is taken into our blood, given to the body uses it, exchanges it with carbon dioxide goes back to the lungs, it's expired out, and then oxygen comes back in, and there's that lovely circle between the two. We looked at the nasal cavity, we looked at the initial filtering system provided by the hairs, and then as it's passed over those nasal conchae, so these little fellas in here, so we saw the air goes past those, it warms, moistens and filters because it's a very vascular area. It's also got lots of little mucus cells which create a sort of degree of humidity, which means that when sort of dusty air comes into that area, the sort of particles get sort of trapped, and it makes that sort of nasal mucus. So that was covered in depth. We then looked at the C-shaped rings of cartilage. So one of your course colouring in, you should have seen that the cartilage, so the tracheal cartilage is created by a sort of like a C-shaped ring. The inside of that ring was covered by an epithelial tissue, which helps sort of moisten and filter that air as it comes in. We went down to the bronchi. So here we've got the left bronchi, and we saw those rings of cartilage again. So structured that way to try and keep the airways open as much as possible. But if you imagine something like bronchitis, where that sort of lung tissue, that lung substance, made up of a slight layer of smooth muscle and other sort of epithelial tissue, can get inflamed. Sort of can sort of overproduce mucus and sometimes sort of limit the sort of air getting through to the alveoli. We saw that as the bronchial tree divides and gets smaller, the bronchi change structure. So these little ones down the bottom, you can see there's that sort of blue cartilage is now a sort of a little plate. So it gives the lung elasticity. It allows it to recoil and sort of fit into that tiny space. But it also means that when that smooth muscle that lines the respiratory tract constricts, it can cause problems. So asthmatics may take what are called bronchodilators. So they work to relax that smooth muscle to allow it to sort of give and open up that airway. We talked very briefly, or we didn't really talk about it actually, but one of your descriptors in your book is it asks you to describe the pleura. So if I'm just going to sort of get my little marker out again. So if we look on the left hand side, here are my, let's say here are my ribs, and if you imagine lining that chest cavity, I'm just sort of drawing it skillfully on the left hand side in orange. So lining that chest cavity, we've got a sort of like a sort of connective tissue lining of our chest called the parietal pleura. So we have so in that sort of area where I've just covered in orange, if you imagine that's the lining of that cavity. Now the lungs, I'm going to draw this in green, are covered by another kind of a connective tissue bag. And I'm going to just sort of illustrate that there. And between those two layers, I'm going to pop it in blue, is you have a fluid called pleural fluid. So the outer, the sort of surface of that chest wall is covered in almost like a plastic bag, but sort of connective tissue. The lungs themselves are covered by a bag, and between them they have a fluid. So when the chest expands, the lung sort of sticks with it, and when it recoils, it goes with it too. So it's almost like if you pick up a sort of wet glass up off a table and there's a beer mat stuck to the bottom. It's almost like that sort of surface tension binds them together. But the pleural fluid allows them to glide ever so slightly, so it keeps it stuck and it allows it to glide. So that sort of little pressure relationship between those two, the two pleura, so the pleura surrounding the lungs and the pleura surrounding the chest cavity, is really critical for keeping those lungs open. On the right hand side we've got a knife that pierces the lung, and it allows sort of atmospheric air into that space, and it causes the lung to collapse and recoil. So we look briefly at the ciliated epithelia, and again, so I'll just cover it in blue. So this is what we call a goblet cell, and so we can imagine this sort of spits out mucus into that surface there. But we also have these what are called ciliated epithelium. So these cells have little kind of wavy hairs on the top, and so they sort of tickle that sort of mucousy fluid along. So from your respiratory tract, it sort of tickles it upwards, so you can either swallow it or spit it out or whatever. So that's your sort of body's natural filtering mechanism. So we're going to look into how the air is drawn into the body. There's a couple of questions en route which I would expect you to answer, and I just want you to sort of consider the mechanisms that are allowing this to happen. So I'll sort of guide you through the pictures. I would, with all these, I do strongly recommend you take a pause and just read the relevant section of your course book, so you can kind of put what you're reading together with what I'm speaking about, and then you can answer the questions and gradually sort of piece it all together, because you should be sort of feeling prepared to deliver your assignment soon. So we have this big muscle underneath our lungs called the diaphragm. It's like a big dome-shaped muscle. So what you see in that picture is how that muscle sort of domes upwards. I'm trying to get my camera. Domes upwards. So it attaches to the bottom of the lungs. This is really a critical muscle for respiration. When you see sort of young kids breathing, their sort of tummies make this sort of lovely sort of out and in sort of motion, and this is the diaphragm working. It's the sort of abdominal contents moving forward, because when the diaphragm contracts, it flattens, as you can see in picture B, and with that it brings the lungs with it. So a bit like a sort of bicycle pump, as you increase that volume, so as you will with a bump, you pull the handle out, it increases the volume within that bicycle pump. In our lungs, we flatten that diaphragm, it increases the volume of those lungs. Now what's really critical here is the pressure of that air in the lungs, and it's very finely balanced. So if our atmospheric pressure, i.e. the pressure outside of our lung, is greater than the pressure inside the lung, air will just rush into our bodies. So if you imagine very finely balanced, in picture 1, with the diaphragm relaxed and a small chest cavity under fairly high pressure, as we increase that volume, what's going to happen to the pressure? It should get less. So we increase the volume and it decreases the pressure. So now the pressure inside the lungs is less than it is in atmospheric air. So it rushes in to fill the alveoli. When the two pressures are equal, that's when your in-breath stops. And expiration, that breathing out, is usually just by a sort of natural elastic recoil of the lungs. So it sort of increases that pressure. If we want to increase, we want to force exhale, we sort of make our chest cavities smaller by contracting our abdominal and our intercostal muscles. So we sort of limit that chest cavity, and it causes a forced out breath. So the key to drawing air into our body is those pressure relationships. So your task now is to pause this presentation, and I want you to put in order these phrases that will show how the diaphragm and also the intercostal muscles will work together to allow air to be drawn into the body. So pause the presentation and get on with it. So hopefully you had a good go at it. What you can see in the next slide is put in order. So at rest, the diaphragm is relaxed and domes upwards. Inspiration is initiated by the diaphragm contracting, causing it to flatten. The flattening of the diaphragm pulls the bottom of the lung downwards, and as the lung is pulled downwards, the thoracic volume increases. The increase in volume is enhanced by the chest lifting as the intercostal muscles contract, and the increased volume means that pressure is lowered. As the pressure is lower than atmospheric air, it is drawn into the body, but as the diaphragm relaxes, it moves back into that domed position, and the lung volume is decreased. The increased pressure causes air to exit the lungs. So that's a lovely explanation and could form the basis of a nice description for your assignments. So this picture, you have this in your books. There's a nice picture in your books which I would encourage you to read now. Quickly pause the presentation and just have a look over that, and then I'm going to talk you through the sort of relevance of each of these sort of confusing volumes. So essentially what you have is you have a person breathing into what's called a spirometer. So essentially as we breathe, just a natural in-breath and out-breath, we don't maximally inhale like this, we don't maximally exhale. We just have a sort of natural, sort of easy partial in-breath and a partial out-breath, partial in-breath and a partial out-breath. So that partial in-breath and partial out-breath, normal easy breathing, is what's known as our tidal volume. So almost imagine a sort of crashing of waves coming in and out. So that usually amounts to about half a litre, maybe slightly more if you're taller and bigger. but normally our tidal volume is a relatively small amount. I guess we breathe about what 12, 13 times a minute. You could look it up. Now imagine you take a full exhalation. So now that's happening here. This is what's called our expiratory reserve volume. Now, what you'll have found is that you can't completely collapse your lungs. There has to always be a little bit of air left in the bottom. Otherwise, think about the sort of lung tissue. There's a lot of blood vessels in there. There's 600 million alveoli. So if you scrunched it up and then scrunched it even further and squeezed all the air out of it, you'd bleed to death. Your lung capillaries would burst. You'd just be a mess. So we always have what's called this residual volume left in our lungs. It keeps it open and maintains the sort of integrity of the alveoli and the blood vessels. So that's my expiratory reserve volume and my residual volume. But now imagine I'm taking a normal tidal volume breath, and then I suddenly take in a huge in-breath. Now, the difference between my tidal volume and my maximal in-breath is known as my inspiratory reserve volume. And that inspiratory reserve volume added to my expiratory reserve volume is equal to what's called my equal to what's called my vital capacity. So this is normally what we measure when we do sort of lung spirometry, because we can never fully measure lung volume. So we normally measure vital capacity to give us an indication of a person's lung size, but we can never sort of fully examine it all. So those are the key lung volumes. I'm going to give a little task for you in the next slide, and then I'm going to give you a bit of time to fill out your book. So let me just press on to the next slide. So what I'm going to ask you to do is complete these sentences. So on the right hand side I've got the words, on the left hand side I've got the phrases. I've got a little picture down there that you can sort of reference as well. So your first question is: residual volume represents the volume left in our lungs when we exhale. Our tidal volume is the amount of air moved in and out of our lungs during inhalation, and our is a measure of maximal outbreath after a maximal inhalation, and expiratory reserve volume represents the amount of air you can after a normal exhalation. So pause the presentation, have a work through those. So I presume you paused. I hope you did. So let's just run through. So residual volume represents the volume left in our lungs when you should have put maximally exhale. Our tidal volume is the amount of air moved in and out of our lungs during, should say, normal inhalation and exhalation, and expiration is, sorry. Our vital capacity is a measure of maximal outbreath after a maximal inhalation, and expiratory reserve volume represents the amount of air you can should be expel after a normal exhalation. Yeah, there we go. Yeah. So the next task is to fill page 38 of your workbook. So you should be able to label the lung volumes and give some sort of idea about what each one means. So pause the presentation and crack on with that. So we're now moving on to the last bit, which is the gas exchange. Well, two bits actually: gas exchange and then control of breathing. Quite sort of complex-ish topics. So we've kind of gone from looking at our gross lung anatomy there, in looking at the trachea down to the bronchi, bronchioles, and we're now sort of looking to pop out at these alveoli. So what you see right down the bottom is these alveoli, and the thing to really show is look how sort of vascular they are. They've got tons of capillaries wrapped around them, and then when you look at this little picture on the left hand side over here, I'm just colouring it in green. These are all single cell thick. You can see those little nuclei in the middle of the cells. The little red blobs at the edge are capillaries, but they're tiny cells to allow maximal oxygen exchange. So I'm going to look at that process in a minute. So this picture, it now sort of explodes the alveoli. So you've got these alveoli, look like little sort of bunches of grapes, and then in this picture here we show an individual alveoli that will have air rushing into it in green, and that oxygen will be transferred onto the capillary. Now, what we have on this right hand picture, I'll just pop a sort of orange on there, is the gas exchange that's taking place. So here oxygen is being drawn into the capillary and carbon dioxide is being taken back out. So the alveoli are the primary sites of gas exchange. So let's see if we've got a better picture to illustrate that process. So what I'm going to get you to do, I think it's important for you to have a quick read of your course books, but you can still see that nice little picture on the bottom here, down here. And I think it's also worth drawing your attention to the little table up here, which is from the course book, which shows how inhaled air has got around nearly 80%, well, let's say 79% nitrogen, but we exhale 79% of nitrogen, so we're not taking any of it in, aren't we? So it's a relatively kind of inert gas for us. But oxygen is nearly 21% when we inhale it and 17% when we exhale. So we absorb that portion that seems to have been lost. But you can see we exhale almost exactly similar amount. So the gas exchange is evident from the difference between the air that we breathe in and the air that we breathe out. And the way it moves is that in an alveoli, what we have is as air enters the alveoli, it's under a very high concentration. So oxygenated new air has got lots of oxygen in it. So it creates what's called a pressure gradient. It sort of drives itself into that capillary. So carbon dioxide in our returning blood is quite high, it's in a high concentration. So we have a high concentration of carbon dioxide. But the air that we've brought into the lung is in a very low concentration. So the carbon dioxide in pulmonary blood is driven back into the alveoli. So it's all about pressure gradients and the gas arriving in high concentration at the alveoli being transferred into the bloodstream, into the red blood cells, and then carbon dioxide in that blood being exchanged and being then breathed out. So what I'd like you to do now, I want to read through your textbooks, and then I want you to answer true or false to all of these questions up here. So just pause the presentation, just have a read through, and I just want you to quickly answer true or false. So go. So hopefully you've had a go. to quickly run through the answers, air in inspired oxygen is low. Oh, sorry, air in inspired air is low in oxygen. Well, I would put that as false. It's quite high in oxygen. That's the point of us breathing it in. Air that is high in oxygen creates a high partial pressure. I would say that is true. It creates a high partial pressure of oxygen, which means it is more readily transferred into our blood stream at the lungs. Gases can pass across membranes where a pressure gradient exists. I would say again, this is absolutely true. So remember, the alveoli are single cell thick. They're attached very closely to the capillaries. So gases actually transfer across that membrane. Oxygen is taken mainly into the blood plasma. Well, that's quite false. A little bit will make its way into the blood plasma, but most of it goes into the hemoglobin, which is inside the red blood cell. Remember that iron-carrying molecule. And lastly, carbon dioxide in high concentration in deoxygenated blood moves back into the alveoli to be exhaled. And this is absolutely true. So if you're asked to describe the process of gas exchange, some of those terms are quite nice ways to kick off your explanation for your presentation, and you might wish to use some of them in your course book. So this is just a little summary of what was previously said. So air entering the lungs during inspiration is highly oxygenated. A high partial pressure of oxygen exists at the lungs during inspiration. This is the breathing in. Arterial blood is highly oxygenated and low in carbon dioxide. And aerobic metabolisms at cells uses oxygen and creates carbon dioxide. So the pressure gradients draw oxygen in oxygen into the tissues in exchange for carbon dioxide. So what I'd like you to do is just have a look at that and explain how you'd finish that explanation to include the blood returning to the lungs and getting rid of its carbon dioxide. So just have a little think through about how you might finish my starter then. Okay. Lastly, we're just going to cover the control of respiration. This is literally, I think it's 3 slides. It's quite a complex topic, so I'm going to sort of try and summarize it as best as I can. So what we've got on this picture here, and as I say, when I send these out, I encourage you to sort of nick some of my pictures and use them in your assignments. There's some quite nice ones that I think you can expand upon. So here we've got one that says ventilation and exercise. So it says ventilation rate jumps as soon as exercise begins. So this is that sort of idea that if you just go for a quick run, your breathing rate increases. But it's probably kind of too soon for major changes in gases to really have been registered. So it suggests here that there's probably a feed forward component to ventilation, the way it responds. So it's almost like it's like your body almost sort of predicting that you're going to need an increased respiratory rate. So there's a couple of mechanisms which will tell our brain to increase breathing rate, and I'm just going to cover those over the page. So just hold in your mind that number one is there's this mechanism that almost sort of predicts for us to increase our breathing rate. So this picture is a hugely complex picture, but I want to sort of just summarize the sort of flow chart on the right hand side, and then we'll sort of look at the various sections that do it. So what we've got is we've got a nice picture of the lungs, and we've got a very complex picture of the brain. The thing that you want to get across is that the lungs are just influenced by muscle. So the lungs are a very passive tissue. The diaphragm is the muscle that causes them to be allowed to draw air in. So really that diaphragm and those associated respiratory muscles are under control of our brain. So something will be in our brain that monitors the condition of our body, and it will create a response that increases respiratory rate to match the demands for either oxygen or to release carbon dioxide. So it will monitor the sort of speed that's required to maintain our body in that nice sort of steady position. So we have three things that's gonna tell our lungs to, or our diaphragm to speed up. Now, number one, we have a respiratory center in our brain. I'm going to color that in green. So in our brain, we have a very distinct section for breathing. Our medulla oblongata, and our pons, which is located around here, just next to our brainstem. So this is called our respiratory center, and our respiratory center kind of sets the rate and pace of breathing, and it sort of integrates information from our body. Now, the first information I mentioned previously was from our sort of emotions and our higher brain centers. So this is sort of coming from here. So I'm not going to talk about that massively, but these two bits we might consider our higher brain centers. So this is the idea that, you know, sometimes if you're sort of suddenly frightened, someone jumps out at you, goes woo, you might start breathing faster. Sort of that sort of almost like an adrenaline thing, isn't it? Sort of you sort of feel like you're breathing more rapidly, but you haven't sort of made any sort of chemical changes in your blood. So there's a sort of higher brain influence on our respiratory system. The other two things that tell our brain whether to speed up or slow down is it's really sort of driven by gases in our blood. The most important actually is carbon dioxide, and carbon dioxide, if we let it accumulate, it turns our blood very sort of acidic. So if you think that our body's tissues use oxygen, and in exchange they create carbon dioxide, so an excess of carbon dioxide is something that our brain would be very interested in. So we have what are called chemoreceptors. So chemoreceptors receive information, or they monitor chemical status of our blood. So the chemoreceptors in the medulla, up in our, well, I showed you where the medulla was. We're in the medulla oblongata. We're around in our brain, in here, right in the center. They monitor the chemical status of the blood. So an increase in carbon dioxide will inform our medulla oblongata. Equally, a decrease in oxygen content and an increase in acidity will be sensed in our chemoreceptors, in our carotid artery, and by our aorta. So carbon dioxide, oxygen, and acidity are monitored by chemoreceptors in our body. They tell our medulla oblongata, our respiratory center, about the status of our body. Now, that responds by telling our muscles to either contract quickly, our respiratory muscles this is, or slow down. So the whole integration is done at that medulla. And if you look on this picture, I'll try and put it in blue actually, a light blue. Can you see here, I'm going to just do a big, can you see how that nerve is attached to the diaphragm? So as you follow that nerve up, you can see it has a direct attachment right in to the base of that medulla. So all the things combined, that initial sort of feed forward mechanism when we get scared or a bit excited, the state of carbon dioxide in our body, the oxygenation of our blood, the level of acidity in our blood, is all monitored by that medulla oblongata, that section of the brain known as our respiratory center, and that responds by sending nervous impulses to the muscles, the diaphragm and the intercostals, that allow us to draw breath in. Nice sort of link. So the same thing happens in the opposite direction. Again, the medulla dictates, so when our carbon dioxide levels normalize, our breathing rate will slow, so the respiratory center will recognize that change in blood gas, and it will slow that rate and depth of respiration. So I want you to just read through this section on your own, give me a bit of time off, and what I want you to do, where I've put my little numbers in here, I just want you to find on the diagram where they are. So just so you can sort of visualize where these things are happening. So our respiratory center is situated in the area of the brain known as the medulla oblongata, and is assisted by the pons. So the two things together are known as our respiratory center. So you can see where those are in our brain, number 1 and number 14. Our higher brain centers may interpret emotion and activity levels and instruct the breathing center to alter breathing rate. The primary driver of respiration increases is our blood carbon dioxide levels. These are sensed by chemoreceptors 5 and 7. So actually I think I said the aortic chemoreceptor was more for oxygen and pH, so I might have sold you a wrong one. So these ones are very much about oxygen and pH. So I want you to tell me which chemoreceptor was responsible for monitoring carbon dioxide. And it's going to be this one, the medullary chemoreceptor, number 3. So this is going to monitor carbon dioxide. So all of these chemoreceptors are what are monitoring blood gas and acidity. So lastly, high carbon dioxide levels will make the respiratory center instruct the diaphragm and respiratory muscles down here to increase breathing rate via the somatic motor neurons. So the somatic motor neurons are that direct connection from that respiratory center on to the muscles of respiration. So there. So you can sort of see how dramatic head injuries, for example, within sport, if it's a thing, can actually affect breathing directly. So a really severe injury to that brain stem is going to really dramatically endanger that whole respiratory center. So that's it's quite nice to show that link between the two parts, the relationship between the chemoreceptors, those chemical signals that monitor the sort of status of the blood, how they inform the brain, and the brain respiratory center responds by increasing rate and depth of breathing. So I think that's your lot. So that's the respiratory system in one hit. It's quite a long presentation, but hopefully you've done it in the stages and managed to work through. And now I want you to work through page 37 of your workbook, and at any stage you're stuck, I would say use the Stafford Brown book. It's a really good source of information, or review any of the information on this slide. So thank you very much.